Thank you, everybody, for joining us for the Pharmacy Cannabis Lecture Series. Each session is designed to deliver a small and in-depth dose of cannabis education. I'm Candace Haas, and I want to thank all of our viewers and customers from the Pottery, the Pharmacy Santa Ana, Santa Barbara, and Berkeley for joining us today. We've had over 30 webinars as part of this series, so please consider following us on YouTube and to get notified when we post new episodes. Our guest speaker today is my good friend, Dr. Kenton Crowley. We're very excited to hear from him later today. But let me tell you a little bit about his fabulous career before we begin. So Dr. Kenton Crowley is a pharmacologist. He's a board-certified PharmD through the American Academy of Anti-Aging Medicine. He received his Doctor of Pharmacy from the University of California in San Francisco and completed his residency with Kaiser Permanente in Los Angeles. He also completed a fellowship in metabolic and nutritional medicine in 2011. During his 38 years in practice, he's worked in primary research at the UCI Department of Pediatrics, the Division of Child Development. He was also involved in the development of several drugs, holds 2 patents in neuropathic pain and patents on a buccal delivery system incorporating cannabis. Dr. Crowley is currently the Chief Scientific Officer for Bioactive Solutions in Reno, Nevada, and is also the Director of Research and Development at Silver State Trading Company, a 40,000 square foot permitted clean green cannabis cultivation and production facility. He also supervises California operations for Trokie, California, and the manufacturing of THC products based in California and CBD Trokie products nationally. He also serves as the CMO for the Patient Care Corporation in Huntington Beach, working with patients and physicians incorporating cannabis and disease state treatments and symptom management, whether as a primary treatment or complementary to traditional other modalities. Clinical studies are also being run through this entity with abstracts and publications ongoing. Dr. Crowley is also a member of the Society of Cannabis Clinicians, the International Cannabinoid Research Society, International Society of Cannabis Pharmacists, and the Incubator Task Force for the UCI Center for the Study of Cannabis. Dr. Kenton Crowley is very generous with his time and knowledge. I've had the pleasure of working with him many times, educating seniors and a disease support community events, and I'm really excited to have him with us today. So with that, thank you for being with us, Dr. Crowley.
Thank you so much for inviting me, Candace. So I do need to disclose that I do derive income from the cannabis space, both as a researcher, a manufacturer, and a clinician. We're going to cover a whole lot of information during the next hour. You should all have access to the slides, as Candace mentioned, as well as the references, so that you can go back and review the details that you find interesting or you want to learn more about the topics that we discussed today. My goal is to expose you to this information and then challenge you to investigate this very powerful and safe adaptogenic herb. So I find cannabis to be beautiful. It's a beautiful plant. On the right is a cannabis leaf, and on the left is an example of a bud, or the cured and trimmed final consumable form of the cannabis plant. Oops. There we go. We have a question. You want to move the slide next?
Yeah.
Oh, there, I guess I can't see it. This is a new format, guys, so I'm learning as we go as well.
So if you click on over to polls, we can see that people are already voting on this.
Okay, I can't. All I see is an option, so.
We have about an equal distribution of people that are voting both the right and the left are hemp plant.
Well, let's give the answer then. Even to the trained eye, they are indistinguishable. On the left side, if you guys picked hemp, you're wrong. That's actually Santa Cruz Tsunami, which is a 4-to-1 ratio CBD to THC producing plant. On the right is a hemp cola with less than 0.3% THC. So what is the difference between marijuana versus hemp? And by the way, Candace, I still have the picture on the left is a hemp plant showing on my screen.
Okay.
So I can't see anything besides that. And I'm not able to, oh, there we go. Okay, sorry about that.
No worries.
So both are cannabis sativa L, but they're regulatory terms based on the amount of THC content. So marijuana has greater than 0.3 milligrams per gram of THC in the dry weight of the plant. Remember that today still marijuana is a Schedule I drug, and by definition, that means that there is no recognized medicinal value and it is highly addictive. Now, hemp has 0.3 milligrams per gram or less of THC in the dry weight of the plant. And in the amended Farm Bill of 2018, Congress dictated that hemp and all its byproducts, extractions, and components were now an agricultural commodity, not a Schedule I drug, legal in all states that have approved U.S. Department of Agriculture programs or something equivalent to that, and by default, federally legal in states that have approved programs. Now, cultivar versus chemovar versus strain. Bacteria and fungi have strains, plants do not. Strain is a layperson's term for cultivar, describing phenotypically different plants, typically named by the grower based on smell, appearance, and physiologic effects. Common terms used is sativa, indica, and hybrid. Hybrids are a mix of the two and make up greater than 95% of the flower market today. Examples of those three would be Jack Herer, Blueberry, and Cannatonic, respectively. Now, Chemovar defines the measurable chemical markers in cannabis that allow us to differentiate between multiple plant chemistries and thus the physiologic effects the plant will trigger. Now, the ECS or endocannabinoid system is the biological system that regulates physiological and cognitive processes in our body, and is perhaps the most important physiologic system involved in establishing and maintaining health and homeostasis. Homeostasis for all vertebrates. That is the term used for balance. It's involved in fertility, pregnancy, pre- and postnatal development, appetite, pain cessation, mood, memory, neuroprotection, and inflammation. Endocannabinoids play a role in neurotransmitter in our immune system, mitochondrial function. They have anti-proliferative, anti-inflammatory, and anti-metastatic effects, and is anandamide, which is one of the endocannabinoids, is associated with the runner's high. Back in the early 80s, 79, when I was in medical school up at UCSF, we were told that the runner's high was from endorphins, or our neurotransmitters that are natural opiates. Now here's, I'm a Pharm B, so I got to show you the structure. So here's an example of anandamide and 2-arachidonylglycerol, or 2-AG for short, which is the endocannabinoids that our body produces. Every cell, every millisecond, we are producing and removing endocannabinoids in our body while we live. Phytocannabinoids, or plant-derived cannabinoids, here's an example of CBD. They look considerably different in structure, but there's important points that these phytocannabinoids behave with the lock and key system in our body, so that the endocannabinoid system is made up of a system of locks or receptors, and keys, we can say, are the active cannabinoids, terpenes, anything else that will engage with a cannabinoid receptor. So CB1 and CB2 are two of the main cannabinoid-dependent receptors in our body. And the first one that was cloned was CB1 back in 1990. CB2 was finally cloned in 1993. CB1 typically is associated with receptors in the central nervous system, but as well as CB2 in the periphery. But what we find is there's CB1 and CB2 receptors littered throughout our body. There are also cannabinoid receptor independent mechanisms that, like 5-HT1A, which engages with the serotonin receptors, TRPV1, the orphan receptors, which there are a huge number of GPR receptors, 55, 119, and so forth. These are references that if you have interest in looking at this, you can go to. Next slide. Just to give you an idea of the myriad of endocannabinoid receptors involved in pain as we know it from 2019. It's, you know, this we could talk about for 30 minutes, just this one slide alone. Next slide. Now this is a diagram of how the phytocannabinoids are handled by the plant. It's plant metabolism, showing you the acid and the neutral. We'll talk about the acid and the neutral forms. But the acid forms of the phytocannabinoids are what are produced, and the master phytocannabinoid is CBG.
Mm-hmm.
Now, if we look at a freshly harvested cannabis flower, and the left side we're looking at THC, and on the right we're looking at CBD, we can see in the acidic form up on the top, before decarboxylation, and I'll explain that in a second, they have significant therapeutic impact, which may or may not be complementary when we change the acidic form to the neutral form. And that is really important for you to understand, because there are products on the market that have the acidic form of THC and the neutral form of THC. The terpene beta-caryophyllene down in the center on the bottom is actually a terpene that engages with the CB2 receptor. It's an agonist, or it activates that receptor, versus an antagonist, which would block the receptor. Next slide. So there's two main phytocannabinoids that we talk about, THC and CBD, but there is so many others. And we have a question up there.
I just launched the question for everybody to weigh in.
Great.
So the question is, how many phytocannabinoids have been discovered to date? 23, 56, 108, or 157?
So while you guys are answering that question, most clinical studies of cannabis today focus on the contents of the two phytocannabinoids showing here, Delta-9, THC, and CBD. Regardless of the fact that there have been identified, most recently, 147 phytocannabinoids. So we have 44% guessed right. Excellent job.
Yeah.
And now over 500 physiologically active substances we have found in the cannabis plant. Next slide. So we talked about the few cannabinoids, phytocannabinoids that we look at today. This is a heat map on the right side, and I know it's very busy and small print, but on the top of the heat map LCMS presentation, you see light to dark little squares. On the top is 36 cultivars, and on the left going down are all of the different phytocannabinoids that were identified. And just to give you an idea, in this slide, it shows at least 10 delta-9 THC types, 10 CBD types, 12 CBG. So you're starting to get the idea that there's a whole lot more going on in that plant than we get to know about. Typically, most lab assays only look at 9 to 13 cannabinoids. And as of earlier this year, most US-based cannabis laboratories that are ISO accredited, ISO 17025 accredited, they only have the ability to look at 23 total cannabinoids out of 147.
Wow.
So there's a lot of stuff that we're not seeing, and that's why there's so much variability to why 10 different people can take the same dose of the same medicine and have a different outcome. Terpenes are the aromatic substance that gives plants their unique scents and flavors. They are classified by the number of isoprene units, 2, 3, 4, 6, and so forth. The smaller ones are terpenes, then they go to terpenoids as they get bigger, and 8 isoprene units are actually a carotenoid. This is an example of 3 different categories, monoterpenes, and the list there, biterpenes and sequestered terpenes. Cannabis typically possesses up to 200 terpenes out of the 55,000 that have been identified. Now, the individual effects, and many times the terpenes will dominate the subjective experience that you experience from consuming cannabis. Next slide. Now, without getting into the weeds, let's take a quick look at extracts and extraction methods. I want you to be familiar with these terms. Full spectrum is an extract that maintains the full profile of the cannabis plant that was processed. Broad spectrum includes that full spectrum of the plant except for THC, delta-9. An isolate contains generally greater than 99% of a single cannabinoid. Now, the method of extraction will determine the components of the plant that are able to be removed, and that content varies very significantly. So CO2 extraction is one of the most used extraction methods commercially in the cannabis space today, but there is light hydrocarbon. There's alcohol, which is used in almost every operation. And then there's even, you can use water, steam distillation, cold press. You can use oils, butter, all different kinds of things to do extracts with cannabis. And they all show a different profile. Mm-hmm. Next slide. So to give you an example of how much we don't know, here's a patient that came into a San Diego dispensary. He was a male, mid-30s. He was still having 8 seizures a day on 4 different anti-seizure medications. So that's called treatment-resistant epilepsy. And so the family was directed to a CBD THC 3-to-1 ratio, sublingual tincture or solution that you put in your mouth, 40 milligrams per mil. There wasn't any added terpenes or measured terpenes, and it was a MCT oil based derived from a diamond OG cultivar. What happened within 24 hours is this treatment-resistant epileptic patient became seizure-free for the first time in his life, and it lasted for the length of the bottle that he had. They went back, got another batch, and they just asked, I want a 3-to-1, 40 milligram tincture in MCT oil. And that's what they got, but they didn't know that it was from a lemon haze cultivar. Seizures came back within 24 hours. They went back to the dispensary. The extractor knew that they had changed the cultivar, went back, made more of the diamond OG mixture, and that patient again regained his seizure control. If we look at the assay of those phytocannabinoids, they look almost identical if we look at CBD and THC, which would be on the label. Some of the labels may include the acid form of THC and CBD, maybe CBN, and very rarely CBG or THCV. If we get down to these minor cannabinoids of CBC and CBGA, again, we're not looking at 147 of them. We're only looking at, 9, 2, 4, 6, 8, 9. So, and there is a difference there. Is this the cause and effect of that person gaining or losing his control of the seizure? We don't know. Let's look at the next slide. There's a whole lot of reported medical applications of cannabis. How could one adaptogenic herb have an effect on so many conditions? And this is only a partial list of conditions patients use for cannabis today. I've highlighted and starred conditions that we see most frequently in our clinic, and many of these conditions require the use of many pharmaceutical drugs to treat and manage their condition, which really sets up for a potential for a lot of drug-drug interactions. Next slide. So our body, our liver, has 2 ways to eliminate things out of our system by metabolism. It's called phase 1 elimination and phase 2 elimination. Phase 1 elimination is using the cytochrome P450 system. Conjugation is phase 2 elimination, and we're really not going to go down that road. But glucuronidation is the main phase 2 elimination pathway for phytocannabinoids. So more than 60% of prescription medications are metabolized by the cytochrome P450 system. All phytocannabinoids can inhibit cytochrome P450 enzymes. The main families are CYP2C, 2D, and 3A, and 1A. THC and CBD are metabolized by 3,4-A and 2C9. 3,4-A inhibitors will slightly increase THC levels, and inducers will slightly decrease THC and CBD levels. CBD is metabolized by 2C19, but THC is not.
Interesting.
CBD so far has been found to be the strongest inhibitor of all of the phytocannabinoids. And most studies show, and this is most important, there is a myriad of potential theoretical drug-drug interactions. But when you look at the studies, the in vitro work that was done, almost all of the concentrations are super therapeutic, which means they're higher than what you would normally experience at a therapeutic endpoint. And those concentrations typically are 10 to 400-fold higher than we would see ever in our blood. So although there is, if you have 400 times the level, you could get a serious drug-drug interaction between a phytocannabinoid and the elimination of Ambien, for instance. But we don't see it when we're using clinically relevant doses. And that's why we see so few drug-drug interactions. But there are some very important ones that we need to talk about. Here's a little more detail. THC is a CYP1A2 inducer. So theoretically, THC can decrease. Inducers are going to speed up that process. And if you're metabolizing faster, then what we're going to do is we're going to decrease the serum concentrations of those drugs that are metabolized through the 1A2 system. And here's a list of them: clozapine, duloxetine, naproxen. Naproxen is an OTC, non-steroidal anti-inflammatory, anyone could pick up over the counter. Aleve would be a brand name. CBD is a potent inhibitor of 3A4 and 2D6. For those of you on medications that have, we'll talk about in a second. So the 3A4 metabolizes about a quarter of all pharmaceutical drugs, and CBD may increase because it's an inhibitor, so it's going to slow down metabolism. And what happens when you slow down the elimination of a drug? It raises the blood level for a longer period of time. And some of those drug classes include macrolides, which are erythromycin, antibiotics, calcium channel blockers, benzodiazepines, sildenafil, which is the purple pill, in that whole group of PED5 inhibitors, antihistamines, haloperidol, for instance, antiretrovirals, and a lot of people are on statins. So it's interesting to note that atorvastatin and simvastatin are metabolized by 3A4, but pravastatin and rosuvastatin is not. 2D6 metabolizes many antidepressants, so CBD may increase because it's inhibiting those serum concentrations of SSRIs. Prozac is a common thing that doctors prescribe to women for anxiety, and instead of looking at their progesterone level to estrogen level in their body, which is probably more of the cause of that anxiety, they would rather give you something that you don't have a deficiency of, and they'll use an SSRI or a tricyclic antidepressant or an antipsychotic, beta blockers, and even opioids. Let's go to the next slide. So in some of the studies that are out there, I listed the drug, the reference for that study, and then the outcome. So warfarin is a anticoagulant. Coumadin is the brand name. THC and CBD have shown to increase warfarin levels, and how that would present when you have too high of a warfarin level in your blood, or your INR, which is a measure of that coagulability of your blood. When it's reduced, the INR goes up. And so there has been bleeding reported because the person isn't able to coagulate adequately or stay in that safe INR level. Alcohol may increase THC levels. Theophylline, if you smoke cannabis, can decrease theophylline levels. There are a couple studies that looked at different drugs, like Indenavir. Actually, the next 4 drugs all showed no impact when taking cannabis, either as a tea or being smoked. Clobazam is another really important drug interaction where we can see as high as an 8-fold increase in the active metabolite of clobazam as well as clobazam. And that can happen within 8 weeks. So clobazam is used for treatment-resistant epilepsy. Then we have some CNS depressants. THC in the neutral form clearly is the product that causes intoxication, but cannabis can relax you and can be additive to other CNS depressants. Whenever we talk about an interaction with 2 drugs and depressing our central nervous system, a lot of times we automatically think it's going to suppress our respiratory system. So if we mix alcohol and a benzo, opiates and alcohol, these are all CNS depressants. They are additive in their CNS depressant effect, and they are also additive affecting the part in our brain that keeps us breathing. The cool thing about cannabis is it doesn't have a receptor in that part of our brain. So it will never, ever slow down or stop our breathing like an opiate or alcohol or a benzo. Let's go to the next slide. Now this one you can hardly read at all, but I wanted to include it so that you can blow it up on your own computer after you get the slide presentation, and you can look at some of this detailed information that Kristen and Elizabeth, both oncology nurses, they're members of the Oncology Nurse Society and the American Cannabis Nurses Association. This was a poster that was presented at CanMed in September of 2019. And it has some really valuable real-world guidance for anyone with breast cancer. So I would urge you to keep this as a tool to look at. Now, there's contraindications. That means something that's an absolute contraindication would be you would never expose a person that had that condition, like acute psychosis or some other unstable psychiatric condition, to cannabinoids. Most practicing cannabis clinicians adhere to that unless you have a very specific skill set that can work with that type of patient. CBD is again a potent inhibitor of 3A4 and 2D6, and so we can potentially impact arrhythmias. So someone that has arrhythmias, THC in particular, can exacerbate that, because one of the physiologic reactions to THC in our body is initially to increase our heart rate. And if that triggers your arrhythmia, then you can have a real problem. Let's go to the next slide. ProjectCBD.org is a fabulous site to refer to, both for a patient or a healthcare provider. And I strongly recommend that you visit this site and learn more about the science, the real life science of what's going on, because they cut through the theoretical, although they can show you that, but they bring it down to real world outcome information. It's also available in Spanish, Portuguese, and Japanese.
Very cool.
So the next slide, looking at reported adverse events, many of these are related to the dose used and the content of the cannabinoids and terpenes. Some of these conditions are actually treatment endpoints, like insomnia, or anxiety or panic attack. Next slide. Now, factors related to adverse events, the dose used and the content of the cannabinoids and terpenes are influenced by the formulation, by the route of administration, the experience and tolerance of the individual, comorbid conditions that can influence metabolism, absorption, and other things like that, drug-drug interactions. Very important, the genomics of the individual, and really where medicine, for individualized medicine, genomics is going to be giving us the answers that so far Western medicine and medicine mostly around the world is just now engaging in to direct therapies. Whether there's product contamination, heavy metals, pesticides, or some other product that isn't on the label, like misbranded or mislabeled products, wrong strength, nothing in there, it's all over the board. So knowing what to use and how to use it is the key to avoiding almost all adverse events. In our clinic in Huntington Beach, we can do that because we've got enough trial and error experience to have a good handle on it. Next slide. Now there's a number of different routes of administration, from the inhalation. Oh, let's go to this. What's the difference between sublingual and buccal administration? So go ahead and make your choices.
So if you guys think that you know what the difference between sublingual and buccal administration is, go ahead onto the poll section and cast your vote.
And so the correct answer is B, sublingual and buccal is absorbed through the oral mucosa. So anyway, there is different routes of administration, and we'll go through that. Next slide. These are the general guidelines surrounding route of administration. So we look at inhalation, which is smoking, vaping, or even a MDI, that's a medication dispensing inhaler. The onset is typically less than a minute, and the duration can be 1 to 3 hours. It's not as anti-inflammatory as all the other routes of administration can be, looking at inflammatory markers in our blood like cytokines. Edibles, I like to consider that group as brownies, cookies, candy, gummies, food, drinks, things like that. Typically 45 minutes to 4 hours of an onset, with a duration of 6 hours to over 24 hours, depending on the dose you take and your GI function. There are many variables here, both between people and then actually the state of what your GI tract is in at the time. For instance, it's dependent on what's in your stomach at the time of administration. Is it empty? Did you just go out and have a steak and lobster dinner with your new date? and then decide, let's finish it off with a brownie or a cookie. And 1 hour later, you don't feel anything, so you eat another one. An hour later, you don't feel anything, and you eat 2 more. And in 4 hours after that stomach decided to empty the contents of that heavy, fatty meal, you all of a sudden got all of that THC, and that edible puts you into a very horribly uncomfortable position. And so when you talk to people about adverse events, many times they'll talk about the edible experience of the candy or cookie or brownie that they consumed, and all of a sudden it just all whacked them, and they don't want to ever experience that event like that again. It's unpredictable as its onset and duration for most people. Sublingual, which there's formulations using tinctures, fatty acid solutions like olive oil, coconut, MCT, and so forth, have an onset of 15 to 45 minutes and a variable length of time that they work. And it really depends on the vehicle and the formulation that is being utilized as to whether you have something that improves absorption through the oral mucosa or not. The other thing is whether you hold it in your mouth long enough before swallowing. So many people on a survey that I've done asking bud tenders in their dispensary, how long do you hold a tincture in your mouth? Oh, 30 seconds and swallow it. Well, if you talk to a doctor or a pharmacist, they tell you 5 minutes to 15 minutes before you swallow. So what you've done is you've changed the bioavailability, the onset of action, the duration of action, all by changing the way you took that delivery system, that formulation. So you can decrease the amount that you absorb if you swallow it, because bioavailability drops when you take it orally versus leaving it in your mouth for a long period of time. The onset of action is going to be quicker. Many of the products, tinctures, are misbranded in the terms of the pharmacopoeia that pharmacists use, the U.S. pharmacopoeia. I think all of our regulatory individuals that set up these statutory guidelines on definitions of dosage forms looked at the High Times pharmacopoeia, unfortunately, instead of the USP. Buccal absorption and sublingual absorption are defining locations in your mouth. So sublingual, under the tongue, buccal, between cheek and gum, and we'll talk about a patented delivery system here in a second that goes under your upper lip. The onset with buccal typically is 3 to 15 minutes for an onset of action. So next to inhalation, it's the fastest way you can get it in your system. Next slide. So here are some examples. This is a calibrated delivery system that is used for inhalation. Here is, go to the next slide. I think it's, so we lost, this doesn't allow animation, Candace. So these are different delivery systems used for inhalation. On the left is ground up, dosed, specifically cannabis flower that would go into a device that would vaporize. Down below, the hands holding a volcano, and on the far right is an MDI delivery system. Next slide. This is an example of why there's so much variability. This is 6 patients that was given in a volcano, which is a vaporization device, which is actually the device used most commonly in studies evaluating the inhalation of cannabis. And you can see from just those 6 people, Cmax, which is the blood level that is arrived at the maximum blood concentration, Cmax was 6-fold difference. That's 6 different people with a blood level at the highest of 30 nanograms per mil versus 180 nanograms per mil. Why is there so much variation? Well, it's because of our genomics, because the dose was controlled, the volume of smoke that was inhaled into the lungs was controlled, the length of time the breath was held was controlled. And so we have all of those variables controlled except the genomics of the individual. Hugely different. Next slide.
Interesting.
Edibles, here's some example of them, and you can look at them later. Placement in the mouth. Typically, if you're looking at saliva production, so if you put a tincture in your mouth and you hold it without swallowing, you will start getting a lot of saliva building up. You know, you're going to be looking like a chipmunk with a mouth full of nuts by the time it's 5 minutes to swallow. There are other delivery systems that go between your cheek and gum, and if the least amount of saliva produced is under your upper lip. If you go back on the top side, or you go down on the bottom side, you continue to make more saliva, and the most saliva produced is under the tongue. Here's an example of some misbranded products, as well as one of the first. This is Sativex on the bottom right of the slide. This is the GW Pharma's FDA in other countries, still not approved in the US, a 1-to-1 ratio approximately of CBD to THC. But the strains in the Driftaway list cannabis-infused tincture. Tinctures are actually a ethanol-based solution, not an oil-based solution. This is an example of the Trokie Rapid Relief Tab. This is something that I developed and patented, and it's a solid lozenge. It's buccally administered under the upper lip and gum. It's standardized dosing, so 10 milligrams per serving, 5 servings per tablet, and this has some very unique properties. It has an onset of action as little as 5 minutes and a duration of action of 4 hours to 8 hours. Something very unique on the market. Next slide. That shows what the commercial product looks like. And next slide. Then we have topicals, transdermal patches, creams. The transdermal patches can be an adhesive matrix or a reservoir matrix. There is 1,000 different things that are out there that you can use. Many of these products work very well to help patients with chronic pain, osteoarthritis, so forth. Next slide. So what is a good starting dose? Most people naive to cannabis should begin with CBD. That's what we learned in our clinic, and we have almost no adverse reactions on starting a person that's never consumed cannabis with our protocol. We upregulate the endocannabinoid system. We're using CBD for the first 1 to 3 days. We get to find out if, for instance, they're using it for pain or anxiety. Is this working or not? Is that dose working or not? Do we need to raise it up? But we start low and always go up slow, because you can always add more. But when you've had too much, especially if it's THC in the neutral form, you can become feeling very uncomfortable. It's important to, and I hope I've described enough to make you aware of the fact that the different delivery methods and formulations have a profound impact on your experience. Pay attention to that CBD to THC ratio and whether it's the neutral form or the acidic form or a combination of those, so that you're not grabbing the same dose of an acidic form of THC and then you take a dose the same size of a neutral form of THC. Entirely different things are going to happen. CBD is the antidote to the THC psychoactivity. So if you get too intoxicated, a 50 milligram dose of a Trokie, within 15 minutes or less, you're going to come back to life. Now, I always like to mention laboratory testing. Testing varies from state to state, so you need to know what they're looking for, but most states are using or requiring ISO 17025 accredited labs, and they're there to make sure that what you're getting is safe, correctly labeled, and contamination free. Next slide. And the next slide. This is an example of a certificate of analysis. You should not buy any cannabis product, whether it's from Amazon or from a dispensary like pharmacy, that doesn't have a specific C of A for that product that you're holding in your hand ready to consider buying. Everything has to have a C of A or walk away from that product. It's looking to tell you what the cannabinoids and terpenes are, whether there's pesticides, heavy metals, whether there is microbial contamination or mycotoxin contamination. This is how we know that the product is safe and why it's so important to go to a reputable retailer that is permitted, because they have to follow all the rules or they won't be in business. If you go out and buy some from your buddy, it's a whole different animal. I like to provide some information sources and organizations that are experienced in the cannabis space. I included a link to Dr. Bloch's presentation on the endocannabinoid system and immunomodulation, which COVID is a big deal today, and the graphics that he's got in this showing how the receptors work is beautiful. So I highly recommend taking a look at his 30-minute presentation. And thank you so much for your attention.
Yeah, thank you so much. I always enjoy presenting with you. You're such a wealth of information. You're so generous with your time, and you really want to help patients. And you can definitely tell that you have a passion for this. And so just thank you so much. We have some questions if you have some time.
Yes.
Okay. So somebody's asking, would you recommend putting an FSO, a full spectrum product, between your lip and your gum? Full spectrum oil, like a Rick Simpson oil or something?
Well, if you're using an RSO or Rick Simpson oil, there's several things you got to consider. First of all, if you're not experienced with cannabis, that is risky if it is a concentrated product. Most RSOs are around 600 milligrams per gram or 1 ml. And when they say take a half a grain of rice, well, that grain of rice could be Calrose or long grain or some other, So there's a lot of variables. Take a half a pea, take a pea size, take 0.05 mLs. That's a calibration that you could get a little better measurement and know exactly what your dose is. But and RSO is typically rubbed on the gum. It can be put into a brand and swallowed. It could there's a lot of different ways you can consume it. Our clinic is seen, especially working with cancer patients, where we're shooting for doing a modified Rick Simpson protocol of 600 to 800 milligrams of cannabinoids a day for at least 60 days. That's a lot of cannabinoids, and taking RSO to meet that goal typically will cause anorexia. You won't be able to eat. You'll lose your appetite, and that's not something that a cancer patient wants to do. Mm-hmm. So you'll find people that love the taste of cannabis, love RSO, have no problem taking. I worked with a guy that took 7 grams a day.
Wow.
He had metastatic cancer that was resolved. Mm-hmm. He was alive for, remember Taras? I don't remember how many years he had cancer, that he was cancer in remission. But he took 7 grams a day of an RSO, and he was perfectly with it. Just like someone on 150 milligrams of morphine or more a day, 300 milligrams. When you get tolerant to it, you can take a whole lot and still operate well.
Mm-hmm. Very good. Would you consider a capsule an edible product?
If you look at the regulations that for each state, in California statutorily, capsules are defined as a concentrate. I look at anything that you swallow as an oral route of administration. And typically, unless it has been modified by mechanical or chemical means, making an esterified form, nano technology to make the particle size small so that it's freely diffusible within our body, which greatly increases the bioavailability of that. Typically something swallowed, and it's under 16% bioavailable, down in the 6 to 12% range. So yes, capsules would be considered inedible, but I try and separate the two out.
Very
Food,
good.
drinks versus regular pharmaceutical dosage forms.
Yeah. And drinks are a new emerging category as well. And people seem to like the drinks because they have a quicker onset and a shorter duration. What do you think about the cannabis drinks that are on the market?
I think some of them are great for people. Especially, if you can, there's some gummies out there that have an onset using nanotechnology of a few minutes. That's an edible. So again, formulation, huge difference. Most nanotechnology products are not real nano. So there's people that do it very well, and there's a lot of people that don't do it well. I have a Malvern in our lab. Malvern is the FDA approved device that looks at particle size through using different lasers looking at particle size. And we, in testing, there's products that are under 10 nanometers in size, and they stay that way for a year on the shelf. And there's others that don't even get close to that. They're in the 100 to 700 nanometer size. So that technically isn't nano. Anything less than 100 nanometers is in the bracket of nano. But when you're down under 10, that's really where nanotechnology works. And definitely the pharmacokinetics are different. Onset's way quicker and duration is way shorter. So you can take a few gummies, get a hard hit with THC in the neutral form, and go to be at a concert, and after a couple hours, you're back good to go to drive home.
Yeah.
And if you need a little CBD to rescue you, then you got it.
Yeah, good point. We had a really good question just come in right now, and this is something that we were talking about just before we went live. And the viewer wants to know, can you generalize about the possible interaction between sleep sedatives such as Ambien and gummies that are designed to improve sleep? So should somebody be combining Ambien and like a gummy with CBN in them?
Well, people do that. It really depends on the challenges of what is causing your insomnia, because there is a very long differential for ruling out what is causing it. It can be lifestyle, it can be a physiologic problem that like pain, for instance, that is uncontrolled or poorly managed. You can have anxiety or some other neurologic issue that is making your mind race, keeping you awake. You're a televisionaholic, because of COVID, and you're afraid to go out, and the television is on 20 hours a day, and you're getting too much of this light energy that affects sleep. It causes disruption. So there's a number of things. Is it a risk for you to die? No. Will it improve the ability for Ambien to keep you asleep? The right formulation definitely should. So, and again, it depends on you.
Yeah.
And again, the formulation, the route of administration, and so forth. You may find that you can go to sleep with Ambien, but Ambien alone, you wake up in 4 hours, even in the controlled release form. And so many people will have a indica dominant cultivar vape that they take a hit on, and in 15 minutes they're back asleep. They've taken something else orally, and that oral combo of melatonin and calming herbs and THC and CBD could either or could help that person go to sleep. So what we find is THC, many people need to be in the 15 to 30 milligram dose of THC in the neutral form in order to get sleep. Sometimes a THCA, or the acidic form of THC with calming herbs, works very well for people. CBD alone can work for people because it is an anxiolytic, it's a muscle relaxant, it's a painkiller, it's anti-inflammatory. And so all of a sudden we got this one phytocannabinoid that is hitting all of these pathways, all of which are not helping us go to sleep. And CBD alone works for people. CBD with melatonin, CBD with something else.
Very good. So what are your thoughts on CBN? I've heard a lot of debate back and forth. There's some researchers that say that CBN isn't really connected to the brain the way that they think that it would actually cause someone to become more sedate and get better sleep. But then at the same time, we see a lot of products adding CBN and saying that these are going to help you fall asleep. What are you finding in your research about CBN? Do you see like any link between sleep and CBN?
I haven't seen much data on that. I haven't actually looked critically at CBN personally, so I do know that it has a intoxicating effect on me. Mm-hmm. So CBN is can be used in manufacturing as a byproduct of decomposing THC. So if it's processed through supercritical CO2 at too high of a temp pressure, then you can get more CBN than you want, less THC. It's an isolate. There are at least eight minor cannabinoids that are available now. In isolate form, so you can make a CBD, CBN, CBC, CBG product, and I've been playing around with those for a couple of years, and they have some therapeutic value, definitely. It's just how many combos can you put together,
Yeah. Yeah, and all this will come with more research, with peer-reviewed studies and everything that we'll eventually get to. We'll be able to really nail down what all these cannabinoids do and what levels you need them, and then match that with, testing every person individually to see what they're lacking. And just like you said, so much of it depends on each individual person and not on the cannabinoids. So that's something that we have to look forward to in science, hopefully sooner rather than later.
Absolutely.
So you talked about it briefly, but I know this is something that a lot of people ask about or worry about. So what are some things that people can do if they're experiencing negative side effects?
Well, depends on what the negative side effect is. So
like if they're feeling paranoid, too much THC,
right.
like uncomfortable feeling is basically the main one that people don't like. I mean, being tired, being hungry, they can deal with.
Right. It's the generally where the problem comes in is the intoxication and the sedation. Anytime you take a therapeutic dose of cannabinoids, it's always drawing, and you probably will have red eyes if it's a good therapeutic dose. You can't get away from that. But CBD is the antidote to the intoxicating effect, the paranoia that comes. I mean, you can even get. I've had people claim that they hallucinated on very high dosing of THC. So it's using CBD is typically the reversal, the rescue. You can use other pharmaceuticals, but it's harder to get your hands on that.
Yeah,
CBD is easy to get a hold of, and it works well.
People can also eat. I find that when I eat food, it will sometimes bring me down. Like you can drink, stay hydrated, drink caffeine or sugary drink. Also, maybe sometimes just, go with it. Like think about your thinking. Like, why am I feeling this way? This is because of the cannabis. This isn't real. Kind of like harness it. And sometimes turn on some good music, turn on a show, read something, and kind of think about and notice how your thinking is different, and like enjoy that and understand that it's a positive thing and not a scary thing or a negative thing.
Yeah. The space between your ears can turn a bad situation into a really cool experience. Mm-hmm. You just gotta lose that fear.
Yeah. And that's why it's always nice too if people are using cannabis or a new delivery method, because they all are like different experiences. For the first time, it's always good to have somebody with you who's experienced with cannabis so they can kind of help guide you and make you feel comfortable. Like, no, it's cool. Nobody's at the front door. We're good. Nobody knows we're doing this. We're safe. We're, just relax and enjoy it. And hey, let's play this game or put this music on, kind of guide people through the experience to make it a pleasurable one. Totally. And then also keeping, a log of your cannabis use, which product you're using, how much you're using, how it's making you feel. And like reading the labels, like you said, can really help you pick the right product and stay away from ones that are going to put you in a negative space.
Yeah. And then going to the right dispensary with the right staff can really give you some good guidance, and the pharmacy has that staff.
Yeah.
There's some really good dispensaries out there that provide top quality guidance. Some of them actually have on staff, at least before COVID, PhDs, mm-hmm. RNs, pharmacists. So there's healthcare providers that are involved in the cannabis space in some high-end dispensaries where they offer, just like the pharmacies offering quality, free education in order to promote responsible use, safe use of cannabis.
Yeah. And some of the new medical brands, I'm finding that too. They have like a nurse line or a help line. Even some of the vet products or pet products have a veterinary line that you can call like anytime, day or night, and get medical information about how to use it, or if like you have some sort of reaction that you need advice on. So that's really cool. And I like to see that coming out of the industry. Awesome. Well, thank you so much for spending the time on this presentation, for being with us here today and answering our questions. I love working with you, and I love Trokies, and they'll be back on the California market soon. So I advise everybody to keep an eye out for them. They're wonderful. One of my favorite ways to use cannabis for sure medically, and really good for people that are immobilized or, can't take a sublingual, but they need that quick onset, which they're awesome for that. So thank you again for being with us. Thank you to our wonderful audience for sharing their time with us. I hope that we were able to share some information that will help you become better informed cannabis consumers, and that the information that we shared will help you find relief. Until next time, be well and stay safe.
Blessings.